Cell block preparation starts with the transfer of the concentrated specimen cells to a flat-bottom glass tube held within a larger tube. The cells are then layered at the bottom by centrifugation before adding the AV marker and molten histogel. The addition of warm water to the carrier glass tube ensures that the histogel remains liquid. A subsequent centrifugation pushes the AV marker through the gel and concentrates the cells into a layer close to the cutting surface of the cell block. Once solidified, the gel button can be removed and processed for paraffin embedding.
Hi, I'm Dr. George Varsegi from the Cytology section of the Department of Pathology at the Medical College of Wisconsin in Milwaukee, Wisconsin.
And I'm Dr. Vinod Shidham, also from Cytopathology section of Department of Pathology at Medical College of Wisconsin, Milwaukee.
And today we'd like to show you a procedure for preparing cell blocks from cytology preparations with a preponderance of individually scattered cells.
We use this procedure to study the immunoprofile of the cells in these individually scattered cells in the cell block sections, and also for evaluation of the special stains on these sections.
So let's get started.
To begin this procedure, transfer about 0.5 milliliters of the preferably concentrated specimen to a 45 millimeters long flat bottom glass tube.